YouTube, how you guys doing? Okay, so this video is gonna be on tests for identifying gram-positive bacteria. Again, this video, or actually this whole series, will help you get ready for your unknown. And if you're watching this video trying to get ready for micro, because you're about to start your lab, this will be a head start for you because you'll know how to do the tests for gram-positive bacteria, and then you'll know what you're actually testing for, etc. So my last video was know your plates, okay? So that would be this one right here. I can link it above, but the next step is know your tests. They're easy, okay? So I'm gonna focus on gram-positive tests. So the first test that you need to know is the catalase test. It's easy. It tests for an enzyme which is actually called catalase, and the bacteria will use this enzyme to degrade hydrogen peroxide, and then what you're seeing in the test is bubbling from, I think it's oxygen being released. So how you do this test is you get a slide, and when I say a slide, I mean the slides that you use to put under the microscope, but you're not using the microscope for this test, you're just using the slide. So you get the slide, and then you take 2 to 3 colonies. You can get plenty, like you can get more than that, but you only need 2 to 3 colonies. Put it on the slide, and then it'll be in this little dropper bottle, and you get the hydrogen peroxide, and you just drop it on top of the bacteria on the slide. If it does not bubble, then it doesn't have that enzyme, so it's not going to do anything. If it does, if the bacteria does have the enzyme, then you are going to see bubbling. Okay, so that is your first test. Then the next test I discussed in my last video. So that would be this video, and I'm gonna link it above. And then when I link it, it'll have like a little eye next to the link. If you click that eye, it'll take you to the exact spot in the video, because I don't want to waste your time and have you looking where I'm talking about the blood agar plate. So how to do a hemolysis test will be just inoculate your blood agar plate with your bacteria, incubate it. Again, incubation takes one day, I think, at least 24 hours. So you inoculate, incubate, and then you're looking at growth on a blood agar plate. You can either have beta, alpha, or gamma hemolysis. I'm grouping these two tests together because they're basically the same. It's basically a sensitivity test, okay? Where you have your plate and you inoculate your plate, and then you get, based on hemolytic pattern of the bacteria, you get the corresponding antibiotic disc. And I'll put a picture up. It's a little tiny disc, and they're white, and its bacitracin is Taxo A, Taxo P is octokin. Okay, so they both look the same. There's a certain strength of antibiotic. You put this disc where you inoculated, and you do plenty of bacteria on there, inoculate, put your disc on, and discs are so small you need forceps to get them, and then you just tap it in to the agar a little bit. Don't like jam it in there, but you know, make sure it's in the agar a little bit. So that's basically how you do the test. to test for sensitivity to these antibiotics. So it's based on a hemolytic pattern. If it's a beta hemolytic bacteria, you get Taxo A disc. Taxo A is bacitracin. And you remember this by beta has a lot of A's in it, like beta, it's AA, it's A. There's a lot of A sounds. So it's beta Taxo A. Alright, that's how you remember it. And then you just put it on there and then you incubate it. After incubation, if there's clearing around the disc and there's no growth right around the disc, then it's sensitive, meaning it's able to be killed by that bacteria. By that antibiotic. If it's alpha hemolytic, then you get the Taxo P. And it's still convenient because in the word alpha there's a P. So alpha is Taxo P, okay? And again, the Taxo P has octocin antibiotic in it. All right, and then the next test would be how do I say this? The next test involves a Bile espelin plate. So if your bacteria is gamma hemolytic, you're gonna grab a Bile espelin plate. It starts out a certain color, but what you're gonna do is you're gonna inoculate your Bile espelin plate, incubate it, and then when you get it back, if it's black, you got a certain bacteria. It has bile and espelin. These are two different types of substances. Some bacteria can grow with these substances. Some bacteria can't. So it's a test to narrow down to see what you got, basically. So if you get that plate back after incubation and it's black, that's positive. I don't know if it's positive or negative, but it can grow with them ingredients, basically. Next test would be a coagulase test. And don't mix this up with a catalase, it kind of sounds similar, but the coagulase test basically uses rabbit plasma. You add the rabbit plasma, again, this is on a slide. A microscope slide. Just like the catalase test, the coagulase test, grab a slide, get the rabbit's plasma, and just put 1 or 2 drops of rabbit's plasma on the slide, and then you get your bacteria that you're testing, and if it is positive, you'll have clumping. If it's negative, there will be no clumping. And I didn't really mention it, but this has nothing to do with the hemolytic pattern of the bacteria. This is a whole separate test, okay? The last test for a gram-positive bacteria. You would use a TSSA plate, okay? So if you're looking at a TSSA plate with growth after incubation, and it's yellow colonies, then that would be positive for micrococcus. The catalase test, which is using the hydrogen peroxide, the hemolysis test, using the blood agar plates, and then you have your sensitivity test to the antibiotics, and a coagulase test, and the TSSA test to look for yellow colonies on a TSSA. So those are all your tests to identify your gram-positive bacteria for your unknown, and those are all tests that you'll be doing in lab. And if you know them before you start your lab, like I said in the beginning of the video, you have a head start. And if you're preparing for your unknown, I hope this helped you. let me know if you have questions or whatever in the comments. And please subscribe if my videos helped you, because I'm gonna continue to do a couple more videos on micro. I'm basically done micro. I'm about to take my test, my final. So if there's anything concerning micro that you want me to go over, let me know. I might do gram-negative identification. I'm not sure. If you guys want it, then